Thanks, Joe. And thank you so much for having me present at this session. As Professor Hill said, my own research is focused on genetics and biomarkers, and so I handle the human genetics papers that come through circulation. I also handle many of the biomarker papers as well as molecular epidemiology papers. So I thought it'd be useful to just talk sort of at a very high level of things that we're looking for in our genetic biomarker molecular epidemiology papers. And this is sort of applicable to basically many different types of papers. And I put it into three categories. One is impact, one is validity, and one is readability. So in the impact space, we really want to understand what's the gap in knowledge that's being addressed. What didn't we know before the paper that the paper is providing data for? What are the clinical implications of the research? Now, we publish and care about all different kinds of research, from basic mechanistic investigations all the way to clinical research. But we are a cardiovascular journal, and we want to understand how are these data going to eventually, at some point, help us better take care of patients? And then what are the unique or novel aspects of the research? What makes this research study unique? From the standpoint of validity, which really is, even if a paper seems impactful and we know it's going to be read a lot, if it's not valid, we're not going to be interested in it. So with regards to validity, are the results robust? Are the methods well described? Are the statistical methods that are used appropriate? Is the study adequately powered? And we like to understand if the results are generalizable. We recognize there are studies that may not inherently have generalizability, but it does help us understand validity and robustness when there's a study that's generalizable. And then finally, with regards to readability, Darren McGuire, one of our senior editors, really focuses on making sure the abstract is really clear and concise, because so much information can be conveyed in the abstract, making sure that the text is understandable for the general circulation readership. Obviously, not the entire readership is going to understand every type of paper, but to try to make it as understandable as possible. And then having visually accurate and appealing figures, I think, also really enhances papers. So I'm going to talk about a couple of papers. One is a genetics paper and one is a biomarker paper, just to kind of dissect out what's the anatomy of a good GWAS paper, of a good genetics paper. So this paper was looking at genetic analyses of left ventricular phenotypes. So they took cardiac MRI phenotypes in almost 14,000 individuals, and they were trying to identify genetic variants that are associated with different components of the left ventricle, different imaging-based components. And they found several different novel genes that were associated with these left ventricular phenotypes, which that in and of itself was interesting and impactful, because not only did they associate with these left ventricular intermediate phenotypes, but also were relevant to clinical heart failure. They also created a polygenic risk score, so combining multiple variants from this genetic study. And this polygenic risk score had clinical implications because it predicted heart failure. We don't expect it from every paper, but in this case, not only was there biology because of the genes that were identified that had biologically plausible connotations, but also had clinical relevance. And the genetic epidemiology was supplemented with biologic context by using in silico bioinformatics analyses. And finally, with regards to impact, that the large amount of results presented in this paper really could serve as a resource for future studies. And then with regards to validity, This was a very large sample size. Importantly, they had a validation cohort built into it so they could really evaluate robustness of their findings. The results were generalizable because they included a cohort that had diverse races and also UK Biobank. And the effect sizes that were shown were robust across these different studies, and the statistical analyses were well done. So broadly, components of impact and validity from this particular genetics paper. And then in the biomarker realm, this particular paper, as an example of this, this was a paper that looked at metabolomics, they did metabolomic profiling in the blood, and connected these metabolites to white matter hyperintensities that were seen on brain imaging. And, why was this impactful? Well, it was the first metabolomic study of this particular phenotype. They used comprehensive and well-done metabolomics analyses. Importantly, it did have clinical implications because these white matter hyperintensities have been associated with stroke. And what they identified those metabolites had biologic insights. With regards to validity, there was a clearly articulated study design. The cases and controls were carefully selected, and they were able to very clearly describe how they had selected cases and controls. With biomarker papers, when we're looking at these different platforms, these different omic platforms, we want to make sure there's technical validity. And the mass spec that they used to measure the metabolites, they showed evidence of good technical validity. They had multiple cohorts to support robustness of their findings. There was large sample sizes, and so they were able to show they were adequately powered. There was appropriate statistical considerations, including adjustment for multiple comparisons when we're looking at hundreds and thousands and sometimes millions of different features. They importantly adjusted for relevant covariates in their model so that we can really understand unique associations between these metabolites, in this case, with the disease that's being evaluated, that isn't confounded by other things. And they compared their metabolites to clinical models, things that we would use when we already take care of patients. And then finally, with regards to readability, there were clearly articulated goals, and I thought that the figures were visually appealing and helpful in putting the results into context, including putting them into context for people who don't do metabolomics for a living. So with that, thanks, and I look forward to your questions.